Until now, understanding what happens in muscles after exercise relied on speculation. However, advances in cutting-edge MRI technology have made it possible to observe in real time the remarkable changes occurring inside muscles during the crucial 30 minutes post-workout. A groundbreaking 2024 study conducted jointly by Seoul National University Medical School and Germany's Max Planck Institute revealed shocking results. The 30 minutes after exercise isn't simply recovery time, but a magical period when muscles are completely reborn. Scientific evidence proves that how you utilize these 30 minutes can create up to 400% differences in muscle growth rates. Revolutionary MRI technology emergence. While conventional MRI could only capture static muscle states, newly developed real-time functional MRI can track muscle changes second by second, during and after exercise. Using this technology, researchers observed real-time changes occurring inside muscles during the 30 minutes post-exercise in 150 athletes and 300 general population subjects. The 7 Tesla MRI used in the study employs magnetic fields over 4 times stronger than standard hospital 1.5 Tesla MRIs, capable of capturing changes in individual muscle fibers. Special contrast agents enabled real-time measurement of blood flow, oxygen concentration, and inflammation levels. The most astounding discovery was the dramatic muscle changes, beginning immediately post-exercise. Far faster and more complex processes were occurring simultaneously than previously known. First 5 minutes post-exercise explosive blood flow increase. From the moment exercise ended, MRI images captured remarkable changes. Muscle blood flow increased up to 800% above normal levels. This resembles a highway suddenly expanding to 8 lanes. Blood flow increase wasn't simply more blood circulation. MRI analysis revealed previously unused capillaries opening, while vascular networks completely restructured. Reserve vessels normally close, activated simultaneously upon exercise stimulation. Particularly surprising was how blood flow increase patterns differed by muscle region. Areas with higher exercise intensity showed greater blood flow increases, with differences reaching up to threefold. This proved muscles self-regulate blood flow according to their specific needs. 5 to 15 minutes inflammation responses. Precise orchestra. Beginning 5 minutes post-exercise. Precise inflammatory responses started inside muscles. While inflammation was previously considered simply harmful, MRI observations revealed completely different pictures. Inflammatory cells moving precisely toward damaged muscle areas were observed in real time, like precision-guided missiles. White blood cells targeted specific areas exclusively. They performed janitor roles, removing damaged muscle proteins. More intriguing was how inflammation response, intensity and duration varied by exercise type. High-intensity weight training produced rapid, but brief inflammatory responses, while moderate cardio exercise generated gradual but sustained responses. Researchers discovered this inflammatory response was the core signal for muscle growth. Muscles with inadequate inflammation showed minimal subsequent growth, but excessive inflammation also hindered development. 15 to 20 minutes satellite cell awakening. Around 15 minutes post-exercise, new changes began appearing in MRI images. Small dots started appearing around muscle fibers. These were satellite cell activation signals. Satellite cells are muscle stem cells that normally sleep, but awaken when muscles are damaged to create new muscle tissue. This was the first real-time observation of this process through MRI. Remarkably, satellite cell activation didn't occur uniformly. It concentrated intensively in areas receiving the most exercise stimulation, while areas with relatively less stimulation showed lower activation levels. Individual differences were also substantial. While some people showed explosive satellite cell activation from the same exercise, others displayed minimal responses. This resulted from complex interactions of genetic differences, exercise experience, and nutritional status. 20 to 25 minutes protein synthesis machinery activation. 20 minutes post-exercise, full-scale protein synthesis began inside muscles. MRI images showed gradual brightness increases inside muscle fibers. This represented new protein synthesis increasing muscle density. Protein synthesis occurred systematically like factory production lines. First, old proteins in damaged areas were broken down. Then new proteins began synthesizing in their place. Through this process, muscle fibers were rebuilt, larger and stronger than before. Particularly noteworthy was how protein synthesis speed varied dramatically, based on post-exercise nutrition. Groups consuming protein and carbohydrates immediately post-exercise showed over 3 times faster protein synthesis than those who didn't. 25 to 30 minutes birth of new blood vessels. During the final 5 minutes post-exercise, the most remarkable phenomenon was observed. Completely new blood vessels began forming. This process, called angiogenesis, involved branches extending from existing vessels to form new capillary networks. MRI images captured existing vessels sprouting thin new vessels like tree branches growing new shoots. These new vessels represented infrastructure expansion to supply adequate nutrition. To increased muscle mass, angiogenesis speed and scope varied greatly between individuals. People who regularly performed cardio exercise showed active angiogenesis, while those with limited exercise experience showed minimal levels. This demonstrated how regular exercise habits significantly influence muscle adaptation capabilities. Differences by muscle fiber type. Research clearly revealed differences based on muscle fiber types. People with predominantly fast-twitch fibers showed explosive blood flow increases and inflammatory responses post-exercise, while those with slow-twitch fibers displayed gradual but sustained changes. Fast-twitch dominant individuals reached maximum blood flow within 5 minutes post-exercise, but declined rapidly after 15 minutes. Conversely, slow-twitch dominant individuals gradually increased blood flow, peaking around 20 minutes before slowly declining. These differences impacted post-exercise nutrition strategies. Fast-twitch dominant individuals needed immediate post-exercise nutrition, while slow-twitch dominant individuals benefited more from nutrition supplied 15 to 20 minutes post-exercise. Age and gender-based change patterns. Research revealed distinct change patterns based on age and gender. 20-year-olds showed all processes occurring rapidly and powerfully, while those over 40 experienced significantly slower response speeds. Age differences appeared most prominently in satellite cell activation. 20-year-olds showed active satellite cell activation starting 15 minutes post-exercise, while 50-year-olds began minimal activation only after 25 minutes. Gender differences were also intriguing. Males showed explosive, concentrated responses, while females displayed gradual but widespread reactions. Males exhibited strong, localized changes, while females showed overall uniform changes. Hormonal influences were clearly observed. Males with higher testosterone levels showed rapid protein synthesis responses, while females with higher estrogen levels displayed active angiogenesis responses. Exercise type-specific muscle response differences. Muscle response differences based on exercise types were remarkably distinct. High-intensity weight training produced rapid, concentrated changes, while moderate cardio exercise generated gradual, sustained changes. Post-weight training showed strong responses only in specific exercised muscles. Blood flow increases, inflammatory responses, and satellite cell activation, all concentrated in targeted muscles. Conversely, post-cardio exercise showed even responses across all body muscles. Differences between compound and isolation exercises were also interesting. After compound exercises like squats, significant responses were observed not only in prime movers, but also in synergist and stabilizer muscles. However, after isolation exercises like leg extensions, concentrated responses appeared only in quadriceps. Critical impact of nutrition timing. One of the most practical discoveries was nutrition. Timing's critical impact on muscle responses. Muscle change patterns completely differed based on post-exercise nutrition timing. Groups consuming protein and carbohydrates immediately post-exercise showed explosive protein synthesis responses starting around 15 minutes. Groups consuming 30 minutes later showed significantly weaker responses, while those consuming after 1 hour showed virtually no response. Carbohydrate roles were particularly important. Cases combining carbohydrates with protein showed much more efficient blood flow increases and nutrient delivery than protein alone. Insulin promoted vascular dilation and nutrient absorption. Interestingly, pre-exercise nutritional status also significantly influenced post-exercise responses. Exercising after 2 hours of fasting reduced post-exercise muscle responses by over 30%. MRI confirmed muscle changes during the 30 minutes post-exercise present us with new paradigms for muscle growth. How you utilize these precious 30 minutes determines your exercise effectiveness. Achieve more effective workouts through the power of science. For more cutting-edge research information, please subscribe, like, and hit the notification bell. Together, we'll help you achieve your fitness goals through scientific evidence.